So friends, let me share my screen. Welcome back. So yesterday, we were finishing the galaxy, exome sequencing. So yesterday I've done this. I was downloading it, and we could get those data sets, as you could see, paid and data sets. Okay, these two paid and data sets. Now we could get it in my history. Okay, this is about one data set is. What you call a control. The other one is a treated data set, okay, which I've used yesterday. But before which, I tell you, this is how your Bauti2 results look like. Okay, in a Bauti2, you get the alignment, and you see this is how your results look like. The first few lines, I'll just copy paste these particular results. So what Bauti2 does is this is the result that you get. Okay, so since we have taken our FASTQ reads, which is of human, I mapped it with the human data sets, and it says that these were paid. These were the data sets that were done, and 99.94% of the data is aligned concordantly, 0 times. Okay, and it says that total amount is this much, but usually, the alignment rate is comparatively percent alignment. That shows that it is there actually. But you can also check these particular results with other Bauti2 as well. I can also show this other results from the other filters as well. So, for example, here, okay. Here, let me see if there is any Bauti2. It's not here. Okay. This is also not there. Okay. Yeah, this is also not there. But there's only one this thing. Yeah. Okay. So this is how your Bauti2 results look like. So this is how I've simply copy pasted, which means 99.94% is absolutely expected, right? That's how it is. So coming to RNA-seq data. So again, for RNA-seq also, we have these data sets, which is a paid and data sets. Okay, but these are the two different data sets. Paid, this is a control, if you remember, SRX19447548, which I got from Get Data. Okay, and the other one is the other one. So instead of Bauti2, what is the tool that we need to use for alignment for RNA-seq? Can someone tell me? What is the tool? Hello, am I there, Goldie? Am I there? Yeah. Please. Yes, please. Can you please tell me what is the other tool that we use?
His state 2.
Yeah, it's called HiSec 2. Very good. It's HiSec 2. So now here in Galaxy you can go and use HiSec 2. Okay. And here when we chose it, somehow Galaxy is a bit slow. We can choose this data, the paid end data. So here we can use the paid end data set collection, because this is what we have used, and the number for this is 118. So 118 is selected. Okay, and then we use a built-in genome, and our genome is human, so we'll use Homo sapiens. Okay, HG38, which is a human genome 38 build. Okay, it's a built-in genome. And there's a strand specification. All these things for keeping in view of brevity of time, it's not needed. Usually, there are different kinds of strand specifications. FR means forward strand. RF is a reverse complementary strand. Okay, FFR, RRF. Different types of strand specifications are there. So, but when you give unstranded, the orientation is unstranded. Okay, that is what you're trying to give us. So this is a paid and data collection, and it is unstranded. Thank you. Click run to. Okay, and then now you're using ISAT2, which means ISAT2 assumes that it checks the splice junctions as well. Okay, and that is how you get these particular results. And then once it is run, so you simply get the BAM file. Okay, this is how in brackets you're getting BAM file. It will take some time, and then you run what you call SAM tools sort. Okay, and then you run SAM tools MPilot. Okay, until sort that is fine. Even MPilot is optional for RNA-seq, and then you run cufflinks. So let me run this. Okay, it will take about 3 to 4 minutes. Otherwise, we have to wait until again tomorrow. It will finish, I'm sure. Although Galaxy is a bit slow, you can you can do this. Maybe as it runs, maybe we can we'll see and we'll get back. In the meantime, can we get started with Sundar's talk? And please allow me to introduce him. Is it fine with everyone? Yeah. So please allow me to take honor in introducing Sundar. V.S. Sundararajan is my mentor. I mean, I'm absolutely happy, and it takes me a great honor in introducing him. I've known Sundar for over almost 15 years now, more than that. And Sundar is a PhD from Singapore and SANBI, South African National Bioinformatics Institute. He worked for various places, including Kolkata, Singapore. He worked for several companies in Singapore. Currently, he is a senior data scientist working for Government of Singapore, and he has been an advisor for Bioclues and my mentor ever since I finished my PhD. And all the databases that you see in Bioclues website is just because of his wonderful long scripts, long lines of codes that he writes. Apart from that, he has also entrusted wonderful projects to children as well, and to many mentees in Bioclues as well, where he trains them with databases and other things. So today is going to give us an introduction about data science and bioinformatics, and he'll give a demonstration about the Veka tool, how Veka does work on machine learning based approaches. Over to you, Sundar. Thank you so much for your time, and let's appreciate Sundar's time as well. It is 2.5 hours ahead, so it is 8:45 p.m. Singapore time. Okay.
Hi, everyone. Thanks, Prash, for the introduction. Can everyone see my screen? Some of you may be knowing, right? The data science is not, without data, there is no data science. It's obvious. And so we start creating data first. So when we get data, maybe we can download it, or we can get it from someone or from our lab. It can be any kind of data. So we are going to see an overview of what are some of these may be known to you. I'm just going to show. So the biological databases are libraries of life science information in various portals, and collected from scientific experiments. From labs, we get a lot of data. It can be a microarray or protein structure or anything. Published literature. So we get information from literature where people have, other labs have done a lot of work on various aspects of biotechnology or bioinformatics, biology, and then we get through high throughput experiment technology where a lot of companies are now handling a lot of data. They produce huge number of data, and then computational analysis is like silico based, how we can um, able to use the knowledge and then create more data. This is the important thing going on, especially on protein structure and drug discovery and things like that. Because everything we cannot do manually, so we use some portion of data and then map it to the unknown, and then we are trying to use the silico computational methodology to build part of the data. So, what database contains? It can be a genomics, which gives a more gene functional aspect, or proteomics, or which gives structure and information and coordinates, and probably some functions, whichever is known, and metabolomics. Then microorigin is, it's through sequence. It gives a huge number of data by various um, and various scientific methods, and phylogenetic is how we compare things are happening, and we know the family, how things have evolved and things like that. Then localization, cellular and chromosomal, or clinical effects, which is now more medical based informatics are based on the clinical of how things are happening, how the mutation is happening. Even for COVID, we have seen it has muted so many times. Very often it is mutating, and then we are surprised to see it's still alive and it's still making a lot of noise in everywhere. Even recently now, I hear that it's also muted, and we are getting more people infected. So, how we do the biological sequence and structure similarities? So, this is a similarity based thing to get knowledge about whatever lab has given us data. So, the database can be on any of these. Even more, I've just listed some of them. So, we get data in a different format, different laboratory, or different portal. So, the purpose is to collect data which are there, and we have to see whether we are able to get a clean, useful data, and we have to match what we want to do with the data. That's our main purpose. So, what are the type of data available? Metadatabases or huge raw databases, which is just collected for huge storage, and organism database or nucleic acid database and protein database, or various databases are available which are coming up, and specialized databases is specific data which we are able to build on our own. As Prashant mentioned, we have even school children, they are able to. I will show an example where the children have contributed 3 to 5 databases, and still we are going to submit them. We are sorting out the server aspect, then we can publish them. So, what are the sub database? Mean the biological database is a big thing. For example, we want to have a COVID-19 data with genome structure and clinical trial. So, what we can go? We can collect this information from various sources, and then we can build up our own database, a simple database, which later I will be showing. Um, that can be used to specific for a COVID purpose. Similarly, we have earlier developed the antimicrobial, which is collection of all antimicrobial things like antibiotic, uh, various sub problem of antimicrobial thing we developed earlier from protein data. So these data is a sub data is created for a specific purpose, so that those because why is it difficult to create such data? Because we have to curate them, because we get If you say antimicrobial, we may get 5,000 records. But do we know that all of them are truly experimentally validated or not? So that is a very big task. Even for some protein, they published maybe 50 to 100 papers. So we have to read all of them and understand whether it is really declared as particular part of that. Then only we are taking it to a database. So that's why building a database is not very easy. It's not like a just dump from some sources and then clean and put it in our database. So we have to curate them because that's the purpose of sub database. So that any new researcher want to compare their data, which is built by them in their lab, it's easy to come to our portal and then check whether it is antimicrobial or it is COVID. And some more we are doing is Nipah virus, things like that. So building our sub database is a big challenge because we need expert knowledge and also technical skill to get the data, clean them, put them properly, and then put it into the web server, which is accessible to everyone. That's a challenge. Any question on this? Okay, we can have the questions later also during a break after every session. Okay, now after going through some slide, somebody thought, okay, I'm going to create a database. So do we need to uh, check whether we need to create it or not? That's an important thing because many people just start and later they realize the such a database is already available and they need not waste their time actually. So it is not good always to recreate a DB unless like some of the database we created. So we can create it back with updates and more tools and everything. But for other people, start from scratch, it may be very difficult because it takes like 2 years or making the data. By the time the data is also becoming old. So it's like a cycle of problem. So we need to have automated coding so that every time we want to run and get the data should be faster because manually building a database is not feasible or it takes longer time. And also we need to bother about the data quality. And first you should know the audience. If you don't know the audience, then building a general database doesn't serve anyone. For the antimicrobial, there was a huge number of citation which was used in many databases because the concept was very clear and good. And so people use them. Even now, people are using and citing the database after 10 years because it's really done in a nice way with more effort. It took almost 2 years to build, to curate the data first and then build the database system on the web server with so many tools. So create a DB as per the user point of view so that the user can understand it. It's not like I create a data, put it in a way people cannot understand. Many databases we say which are not very clear. Uh, the moment we see the menu, we should be knowing what we are looking, where we want to search, things like that. Otherwise, it becomes like something like a Unix level where you have to put one by one command and see what is coming out, rather than the graphics user interface where but a click of mouse we are able to understand what we are looking, what are the submenu and things. So create it when the user is able to use it and create simple search engine. Don't complicate things. Write very big coding and it gets stuck in a loop. It will never come out. So clarity and easy access because make it simple and everyone can access it. It should be like a easy page wise. It doesn't give everything together, nor I can go first page or second page like that. It should be easy access so that it becomes more easy to go through the full data as per the slow or page wise, something like that. Open access. This is what Prashant is fighting for last 15 years. That people put everything and then say you have to buy this by paying dollar or something. Because we should support open science so that more new bodies can come. They are able to understand and collaborate, and they also come up with better ideas. Publish and give more details of usability to researchers with some note or how to use it, things like that. And 10, last not, but not the least, most of the database die because we employ some researcher in different level. They take over the database and complete it. Once the PhD or a master completed, then no one is coming back to hold the database because immediately one cannot take in the next year to publish because minimum 3 years are required to republish or upgrade databases. So meantime, the database become old by 3 years, and then it's not useful. Even my antimicrobial is died already because where I work, that server crashed and they never buy new one, so it's still dead. Now we are going to recreate, and maybe some of you can work with us for some of the databases, so we are able to put it back, and then we can publish it in the NAR. NAR, as you know, it's going to be a almost 20 impact factor. We can try next year, but now is already over the timeline for this year. So maintenance and update is very important. At least we suggest every 3 months we need to update. So as I mentioned, when we update, we should see that we are not doing it totally manually. It's difficult to manually cut and paste or process it. So, but then we need to code it. We have to use Python, we have to use PHP, we have to use MySQL. All these components are embedded. So when we get a new text data, later I'll show, we can update the database instantly. I've developed a lot of tools, so that helps in updating the database immediately within 1 or 2 hours, and then we can republish it or update it very easily. Okay, these are some of the very simple things most of you may be knowing. So what is data? Can some of you tell what is the data? From A1 to A9, which are called data? Maybe you can put it in your chat box.
Sir, everything is a data. All information is data.
Anyone not agreeing? Okay, everything is data, right? Now, whatever you call, whatever you say, whatever you visualize, picture or anything is data, because now the data is scaling up like anything. Now, we are doing now like image, not only image, coordinates and pixel level. Everything is going on data and comparison and analyze them, how things are happening. Graphical network is going on. The entire graph can be networked to compare the graph to graph and build network, things like that. So everything is data. And then this is a simple thing. If data is in tabular form, first column we give the student name or then mathematics or science. So this is a simple format to tell data is divided row and column. So when we put it in web server, it's also same. We need record by, like student 1 is 1 record, and we give maybe 2 or it can be 200 columns, all the information we want to hold for a particular protein. Like protein 1, we may have a lot of information, where it is published, who published, what are the author, where it is published, which year, journal. So when you break, you can see there are huge number of information. It can go more than 1,000 columns. So all that we need to break and put it in a data format. Then only we can put it in the web server. So that's where it's very important to break the data into column wise. So what is data? Data is a datum that comes with facts and statistics. So data is a plural form of the word. It brings facts and statistics. So what is a table? Now we go next step. If you have one table, which means this is a student table. But if you have two tables, that means the student details can be in another table. So we get two tables and then we are able to combine them. See, so when we have table, we see it's a table. So what is a database? Database means we have more tables. We are handling more tables. So one can ask why we need more tables. See, student one, we have mathematics. Student one can repeat because of term exam, exams are coming. So that list can grow and there will be a repeat in the first table, like student one repeat second. But whereas address doesn't repeat because student one resides in one place until he changes his address or contact number is changed. So it is only one time. So if you combine everything, we need to repeat data that many times. So which is redundant. So we don't unnecessarily waste our data because later things grow, data grows bigger. So that time we don't have space or we cannot manage it in the server. So better is to see whichever table can be divided with the uniform information, unique information. And whichever grows, that will be a simple table just growing several thousand rows. So these are table are divided and then it is merged. So when I take all information, so it takes from student one resides here, mathematics, science and other subjects. So everything is for the name, everything in one page is combined and we get that data. So the same idea will be done in web server also. When we launch it, we have to carefully plan what are the tables we need and what are the data we need, unique data or repeated data. And then build the columns, have the columns or divide them in a proper way and then have the table. Later we combine them and put it on the screen for the user based on the user requisite or what they want to see. So these are the Uniprot where we usually get a lot of data because it's more clean and directly we see most of the records are experimentally validated. Some are still not validated. So we can think whether which part of data we can use fully experimentally validated or all together. But still Uniprot gives a more clean data on. So usually I build all my program on Uniprot only so that it's very clean, easy to handle. And another one is major one is NCBI, but NCBI data so we used earlier, but it's a bit complicated because the format is not uniform. It doesn't have a row starting. Later I'll show. Every row we cannot know whether it is a continuation of the previous one or a new row. So they don't put a tag in the first column, first one or two column to say what it is coming following it like that. So that is a problem of it's like a very pure unstructured data text format which is very difficult to handle by computer. But this one gives a coded thing. First column gives if it is author or source, where from accession number, things like that. So it first two column give everything information what is followed. And this one doesn't follow like that. So we have to manually understand what they are telling on each column and then to manually divide them. So that is a quite challenge. And also NCBI, we can see a lot of repeats will be there. Whoever just dump, they just dump it. It is not that same like COVID also you can see huge number because the same thing done by various lab, they just dump it. So sequence will be repeated. So there is a trade between these two. If you take NCBI, you can get it yearly data. But if you take Uniprot, it is comes a bit late, but more refined and clean and clear things are coming. So that is a tag between them. So what are the some tools I put here? So programming, we can use Python, Java, C++, but Python is the one which is going very well now. On RDBMS, you can use Oracle if the office has Oracle. Otherwise, it is very expensive. So we use MySQL, which is a free version. And WebTool, we use HTML, PHP. So all these are used for building a web server. And then system, we can be have Linux, Unix, Mac, Win. There are so many flavors are coming in Unix or Mac or Windows. Okay, so this section is done. You have any question? Because the base is building a database. Before that, we need data. So, any question on this? Hello, everyone is there?
Hello. Good evening, sir.
Yeah, Deepika.
Sir, my question is like you said that on NCBI, the data is like dumped in that way, and we are having many sequences of the same type. So, sir, there is no like filter or some feature which can filter out the sequences which have been already discovered or already present, and the data is not fed what is like being dumped there.
The main issue is different labs at the same time they structure the or construct the data, right? And then they put it in the NCBI. So parallelly, when they do, we cannot know, right, whether anybody in the world they are doing that part. So there is a possibility of repetition, and also like references will be differently put by each, because each one they just put their name and details and then submit it. Not like Uniprot, which is they do a work to combine things. If they have the same common thing, they combine all the papers published under that together and then put the sequence. So it is more easy thing to know whether it is same sequence. Two people are talking. When I see the Uniprot, I'll have more number of publication. Mean they're talking about the same protein and same sequence or whatever they are talking about. So Uniprot is more classified way and collection, which is becoming more easier for us to know that so many publications are done, and then they continue to add up things rather than putting a different entry for the same sequence. So that's why NCBA is like a quick thing. I can just dump it and say I have done it, but without bothering whether anybody else has done. So that's how NCBA works, most of the thing. So it becomes difficult for us to know whether it is already published in Uniprot or not. So manually we have to check. Some IDs still have the connection between Uniprot and NCBI, but NCBI doesn't give all connection. That's a problem. But rather Uniprot gives all the collections with all the 200 plus over databases, and they do things systematically as a big team. That really helps us to know where these are all published, in this, which, what catalog name or ID, or even they give a link also where to go and see. Deepika, if you use the NCBI, then it becomes more challenge. That's the thing. Sometimes we are using hypothetical protein, so we are developing, but some we got about 600 new entries in NCBI. So now it's also we are trying to see whether all of them are correct or not. So it, and then coding is a very big challenge because it's a free text, not orderly done also. So later I will show you how Uniprot is easy to organize by the coding. Is that answers your question?
Yes, sir. Yes, sir. Thank you, sir.
Yeah, welcome. So any other question or we go to the next session? Okay, before we go further, one should know what is big data, because people talk a lot of name these days. I'm doing big data without knowing whether they're doing big data or small data or what they're doing. So we can have a run through on this. So this is a standard picture we see from many places, that how informations are collected. The bioinformatics is everything. either it is database or pathway or annotation, NGS data, genes, DNA, anything you name in the dictionary. It has become a terminology has become a dictionary now for all this, and everything is a data. So it's becoming more challenge. Why? Storage. How much we can store? How much data we can download and store? Transfer. How we are able to transfer it from one computer to another computer, or because our external hard disk is now 1 terabyte. So the maximum is 1 terabyte we can use. We can carry from transfer from one computer to another computer, or OS also change, whether to Unix to Windows and things like that. So how we are going to do analysis? How to analyze the data which you have collected, and how to present it visualization. So these are the challenges we are facing. How we are going to handle the big mega data. And also this is a usual thing, like why data is growing, because the cost of genome sequencing has drastically gone down. That becomes every year we are able to cut the cost and do more sequencing. Now even you can see the COVID, every couple of months new type is coming. People are able to immediately develop it and submit the sequence. Even I see many sequences are repeated because of the various labs. So the other problem we feel is genetic diseases, because on the other hand we have huge information, and the challenge now is how things are connected, correlated between genetic and the diseases. So are we able to understand to gene or protein structure? So this is a challenge we are all facing, and we are trying to get the information so that the cure is going to become easier, and also the companies can create new drugs information. They are able to map between the disease and the genetic information they have. So the big data is if you have a big data, and also we have analytical tools. So the advantage is we can identification of treatment of disease at a faster scale, because we know cancer and all is difficult to identify in the stage 1, which becomes stage 3, and we are unable to save the person. Rarely some people could be saved. So if we can understand things faster scale, then we are able to identify the disease correctly and start the treatment earlier. And cost is reduced. If you have a clear, clean data, then analyzing them is becoming a better option and reduce a lot of cost instead of searching for data. So health is provided at an early stage, and then the data is augmented. Means we have more data and regular updates. In many labs and all, they follow the patient, whether it is cancer or other thing, what is happening, how their health is progress, and so they get data regularly, and then it is added and see what is the problem happening on the downscale, or how the people are living and things like that. So this gives an idea of how things are happening on a disease, on a so this helps also disease control, and people are able to control if it is like dengue or COVID. Sorry. Okay, what type of data is collected? We collect, as we mentioned, DNA sequence, whether it is a full genome or gene, and then amino acid sequence of protein, three-dimensional structure, nucleic acid, or things like that. So there is a huge growth of data because a lot of genome project is happening. And on the other hand, what we need analytics. So bioinformatics and computational biology involve the analysis of biological data, DNA, RNA, protein sequence. So we are able to analyze them and understand the structure, or how to map the function. So that helps in building later the drug how we are able to build a drug. That's the main thing, so that the cure is possible. Okay, let us see in a different way how the impact is happening. So if we take biology, then in the traditional way, we get sophisticated technique, but now the modern technology, we get cost effective, time saving, better analytics. So the big data, like genome and protein sequencing, and we are able to predict protein structure in a much better and easy way once we get all the proper mega data. For the sequence, we have getting big data, building genetic codes. Early genes are mapped. Now the identification of human genetics, and then the genetic disorder and blueprint is able to make, and that helps in reduce costing because sequence are not made fully. They partially make with computer-based thing, prediction-based technology. So 50% they try to predict and then map it rather than repeatedly do the mapping of the gene sequence. So less errors due to analytics algorithm rather than doing it in a lab, which may have a lot of problem in the probe and things like that. Every time they get different set of data. So we may not know whether which one is correct. So rather we go for analytical way of getting data that will become more better and clear and cleaner and faster. So, as we said, now the trend is going for personalized medication. I mean, everyone can have their own medicine built based on their problem, intensity, and how long they are suffering. So that is still not yet ready. But these are all going to happen when we have a big data and we are able to pinpoint who needs what. Then we are going to get maybe, I don't know, maybe 5 or 10 years later, everyone can have their own medicine, which is like how we order food, we say the parameter and we get a food. Similarly, the medicine going to come where it is only for me, that medicine. Only for somebody, the another medicine will be created and given through some vending machine or something like that. So data on genomes and proteomics, metabolomics, all are we have to use, and then we are able to build disease models because that is more important to build models for to know whether a new candidate or a patient is going to have particular disease or not. So we need to have a disease model to get a understanding whether it is tend to have this problem or not. Then only the proper treatment can be started. And structure of disease that helps in getting a potential drug so that the drug binds and things. Most of you will be knowing better than me. So that able to solve the problem of disease by having a proper drug that can be binded and resolve the problem. So the idea is the mega data can help in developing target drugs more efficiently. Because when the data we have, then we are able to analyze it with the tools and analytics and software. Then it is easy for us to move on. Nowadays, you can see the drugs are coming much faster than earlier, like because we get more data readily available. So the manufacturing pharmaceutical no need to wait until they know what they need to have data before they start anything. So that is the way things are moving in a better scale. Like even for COVID, quickly we got the COVID vaccination and also 1, 2, and things are coming. Different company, they're able to make, why? Because of they have the sufficient information, how to move on it, how to tackle it, what is the structure. So all these are because of collection of huge clear data that help them in building the medicines or vaccination much faster. So that's how we saved a lot of people in the world, as a classic example of COVID, which also killed a huge number of population. But on the other hand, because of vaccination, we saved a lot of people on that. Still now, people are getting problem and vaccination is on. But the death rate reduced for those who took vaccination. Some, they could not take vaccination because of some problem. And also still it's a question. So we are able to model it at the right time, and which can help in prevention of disease, disability, death, and things like that. Another dimension, we have deep learning and machine learning. So when data collected, whatever which is established through lab or experimental or clinical or um, we can say silico through computer, all these are huge data. So but on the other hand, we don't have huge data which we don't know what they are doing. So the deep learning and the machine learning, all this can go depth in understanding through the analytics and machine learning technology algorithm that build an idea, gives an idea what this particular data is going to do functionally or medically. So this gives an idea, and this field is growing much faster and finer day by day, so that things are happening. And it may not give 100% thing. If you, I say I get 100% accuracy that this is this, I don't believe it, because maybe some data bias is there, or some odd noisy data is in between that make everything wrong and give a big number. So if anything, later we will know that neural network and some, if I get neural network 80%, then we can say it's more or less a correct way of getting the accuracy. That model can predict more or less properly. And then that tend to be a function or a particular thing. So the deep learning also give a complete domain knowledge. I mean, if the model is not exist also, we are able to map it. That's the idea, and use it for another problem. If I solved one particular type of COVID, the other type, if I don't have enough data, still I can map it and see how things are happening some extent. So that all the algorithm is able to divide, and we are able to understand them. And we have platforms, cloud platforms, where we have a lot of tools and things. So this in turn help better understanding of human health and diseases. So the mega data is helping us. So these are all you can see, go through later. We'll put it in the, we will give the slides. So these are all a lot of papers on this one. So what are the challenges? The business analytics fail to provide insight, means lack of data. So the data is incomplete, and long delay in data analytics means update. Traditional expectation not matched in new model. So all this, what are the business point of view, the analytics, which are getting in problem? Means the expert knowledge is not embedded to the model. Because when we build analytical model, we need the data updated. If it is not updated, then we are not getting a clearer information. So that is a challenge. And the another challenge is inaccurate analytics. Because if we don't have proper insight, then the data may be poor quality, means it's not very clear. Every time I see a particular set of data, I tend to go wrongly and suggest new things. Function is keep changing, and then system errors. It's unable to process it properly, because a lot of data may not be so accurate, and then the model is not accurate because the business analytics is published or established or laboratory through then only it is going to be a accurate model. If we don't have the base data accurate, the model will be totally wrong. So the model we have to check with prediction, whether it is some part known data. We always check with the model to see whether it is the prediction is correct or not. Then we can see how the data modeling is going on correctly with the data. So the data from which we can build disease model, and if we cannot balance it, then we can say it's a big challenge. So analytics, as I mentioned, unclear data visualization. Sometimes the model requires more data, because sometimes for cancer and all, we get very less data and becomes difficult to make a model, because the unknown will be very big, known is very small. Then it's becoming more challenging. We are not clear that whether it is suggesting properly or not. So the idea is to revalidate, and data must be useful, and it's involving external domain expert to decide on data, because some data may be noisy, or we need to add more data before rebuilding the data. So another issue is if you add too much of data, so the system takes a longer time, because we don't know whether the system is working on it or finally it is going to crash, and time delay in getting knowledge, because we don't know whether our data collection can give some knowledge or not, whether we are able to predict something, or we have to divide the uh, total group patients as they are having the disease or not having the disease. We cannot divide them, then we cannot move further whether we are going to do some treatment or not. So the data organization, if it is huge volume and if it is not in proper order, then it is going to be a very mess. And what we can do, we can increase the infrastructure, and we see the resource utilization, or we have to upscale data augment, means add more clean data so that it is clearer and processed properly. And this is a very big challenge, because having more data regularly, we need to have more resources, and it becomes more expensive maintaining them. First year we buy, maybe 1 or 2 years we get warranty, and then it becomes challenge to get money. In most of the lab, they don't get money for maintenance. So then what will happen? It will crash, and then it becomes more challenge to add more data and put it in another machine. So the cost is going to add. So we need to have a better hardware. So we need to suggest cloud solution, because if our data is growing clearly, then we can use cloud solutions which go by pay as you use methodology, because AWS give whatever amount or space we use, they charge. So it's good to do that, so that we don't put more money on hardware which we are unable to maintain later. Rather than we can use cloud solution which is based on how much we use, and also they give a lot of credit for industry and research purpose. I think like 1,000 hours, something like that, they give free for a project, so we can use that also. So simpler version, then we can scale up the system. So anything, any questions? Yeah. Hello, still anyone there? Yeah. So
thank you so much, sir, for your wonderful talk. Before you begin the hands-on, I just have a quick what do you call announcement to all the participants. Friends, I would appreciate if you please come at least join at least 3 or 4 minutes in advance when the workshop starts. We cannot wait for you, and kindly do not share this particular link with anyone. Okay, this is only for the paid members, life members, where all of you have just paid a paltry amount of 2,000 rupees. I'm sorry to say this, but this is how we work. We are a not-for-profit organization, and during the last 30 minutes, I had to deny admission for at least 33 people, which means that some of you have really shared these particular links with, with your colleagues or with your friends, which is not probably acceptable. And sorry to be very open on this, there is no free cup of coffee anywhere in the world. And we at Bioclues work absolutely differently. Okay, so kindly do not take it otherwise. Okay, my time is very precious as yours, and so is Sundararajan's time. Okay, so there is no free cup on the earth anywhere, free cup of coffee anywhere on the earth. So kindly, please be advised on this. Okay, it might find a bit rustic or a bit horrific for you, but it is not a news for us. And we take pride in saying that ours is the largest bioinformatics society where we spend Herculean amounts of time for mentor-mentee relationships. Okay, and we work without any penny. Okay, so kindly be advised on this. So your time is very precious, and so is ours as well. So the workshop time is intended at 6 o'clock. Please be there by 5:57 p.m. Okay. I need not tell you you're all revered scientists, you're all revered researchers, or all revered participants, and we're all scientists working on the working to ensure that the life is we make our lives and other lives better. Thank you so much. Thank you. Sundar, over to you, please. Yeah. Dada, your mic is muted.
Yeah, I'm talking to myself. Okay, good. So, have you all downloaded this Veka software? Because why am I telling this before we will go through a bit on Python. I want people, if you have not done on a one side, you keep downloading this. It takes a while and install it. All of you have downloaded this? I mean, anyone not done?
Sir, I have not yet completed. I'm doing it now.
Yeah. You give it on one side. Now we are going to see what we have done on web server and how we are going to do Python and things like that. Because this one is the easy software, even though we need to understand a lot. But this one is a click of button, mouse. It's become more easy. They are also same thing. If the data is not clean, it won't work. But we have the good number of already published and well curated data within the Veka library. They give a lot of data so that we can see whether we are able to understand them before we really build our own model, because that takes longer time and it's not like a 30 minutes we can understand everything. Okay, so but it's easy to click and play. So meanwhile you download it on one side while we go through the other things. Okay.
Yes,
Okay.
sir.
Yeah. So this is children. The girl is class 5, age 10. She also part of this database. Even we got surprised. She's challenged that she can understand, and she got 3 viruses. She went through and gave 3 viruses on this. So this is ready, but we are going to update this and then submit. We want to add some tools also. So we can see the collaborator children all are in different school, and this is I am talking about Nabia. Now she is in class 6. She is from Andhra. She is the youngest, and she contributed 3 proteins, and others are also there. All only school children. All these I'm dealing only with school children, and they developed almost 3 databases, and 2 more are under the final stage. So we are going to publish them. Already some publisher have agreed to publish all these. Okay, so the data, once it is ready, so what we are doing, we build the catalog. So that's the thing. When we do, so we take maybe because sometimes we didn't check, the server is having some issues. Okay, so it will give, the idea is, see, we can see all this tabulated the data. We are still cleaning and making the children are checking on the data. So these are all the catalog we built with the data which are from Uniprot. So we got 9 entries on Nipah virus, and this one still we are working for the latest data, and then we are going to submit. So the idea is take the data, break it, put it in a proper format, and then upload into the server through MySQL, and then this catalogs will work. So this is how we are designing the database. So how this is done? That is a big challenge. Huh? So how you divide? Okay, so I take one data from Uniprot. So this gives, as I mentioned, accession ID, accession number, date, submitted, description, all the data, and then where it is published, author name, where it is nucleic acid sequence, and things like that. So this gives a way of, as I mentioned in the first two column, it gives what the following things are there. If it is OC, all it's coming, all the classification, the virus, and the similarly, all the comments what they are giving on the subcellular and where all this is connected. So you can see it's all connected with Go database and the same protein connected with CDD database, all mapping in. So all these informations are updated in Uniprot. That's advantage. So we can see how many are added up with this for same protein for the Nipah virus, and then it goes over as published keyword. We get huge number of keywords also, how it is derived, and then what are the other details we got from publication. So all these are added, and finally the sequence. So the Uniprot gives the things in a clear way, and it gives how things are taken out from publication, published material, and things like that. And they sum up everything together from various databases and links, and then we get this is one entry. So all we are seeing the catalog all from this. So when we want to when you want to go author, like we say keyword. So all these are coming from keyword. So all the keyword are, then we know how many times, how many entries are coming. So host cytoplasm is 5 times is referred. So when we click, we get 5 entries from the database. That's the idea. So, and then citation. So this gives all the details, author name, title, who published. So this author, Abu Bakr, has 13 publication. Even though we have only 9 entries, but he got more publication. Means some different publication and aspect on same protein or something he published. So, and journal and year when it is published. So all this we break it through Python coding, and then we create catalogs and then put it in the web. So this gives easy access to author. Any author who want to know more on this, they know which year published or which journal it is published or things like that. So we can see lot of journals are there. Virology 7, Nature also one. See this Nipah virus published in Nature also. So all this information we catalog, all we get data in the same way we process it. Similarly, taxonomy, which gives a classification, but these are all should be same because it's a one family. Whichever way you call, it is 9 entry, whether it is virus or anything, it's one and the same. We got 9. So all taxonomy goes to all because there is no other division is there. And these are all done by children. For only the launching of backcoding done by me. Children helped in checking the things, getting the matter and material, and cleaning the, checking the, because we need people to check. See, just now I see some links are not there because we didn't update it for quite some time. So we need somebody to check. So children do things very clearly. After the school, they come back and say, "Uncle, I am back. Let us work on it." So everyone is very curious and working hard, and they're able to achieve things in a better scale. So this is how we upload to a database. Now we go for Python coding. So, so how many of you don't do coding? Wow, I see many numbers hit. They don't do. Means you do coding, right, Arjun, Ziaul, and Deepika. So you are doing coding? People also. Oh, I mean, even you all don't do coding. Ha ha ha. I was very happy that so many people can do coding, but now I
Sir,
see.
Python we do only with Google Colab. I'm not that much expert to run it separately, so I'm not that kind of person who writes the scripts actually.
Actually, coding is an easy thing, right? It's not a very big challenge, but it takes a while to understand. Yeah, most of you don't do coding, so we need to start some class for coding. That's what I realize, because coding is not a difficult thing. Only thing, it's like how you write a poem or something. If you are creative, then you can write it. Language doesn't matter. Whichever language you use, Python or Java, doesn't all matter. Only the way how we tell to computer changes between languages. Doesn't matter what we are telling to the computer, because that is human. I mean, that is called logic. Human knows the logic. Computer doesn't. Only it read what we are writing. If it cannot read, it will tell something wrong. So, see, this is the coding I have done. I won't give a copy because still we are publishing it with the children. So, this takes the Uniprot entry and break it. And then it breaks all the thing, like it breaks ID, it breaks AC, it breaks what is the organism, and then whatever COC means, it is the taxonomy, and then very simple. Each paragraph does its own challenge, and and then after breaking everything, we get here also the author name, journal. Here, see, we break the journal and everything, and also we do some checking how we get the volume. Everything is automated, but sometimes it may fail because some new line has come a wrong way, and then sequence, how we collect the sequence, and then we build the SQL and then put it in the SQL format, because SQL format has a specific way we have to put, and then we upload to the server. And then these are all the, see, divide year, journal. These are the catalog, how the catalogs are built. The keyword and the taxonomy, all these are in built catalog, then we upload to the server. And then we build the catalog. See, construction of the catalog goes in this way. Don't think this is done just like a easy one. It takes a while. I understand. It's not. So we take this data and see if AC, then the AC is broken here. On one run, it will check where is the AC. It is AC. So this paragraph will work only for this AC. So when AC is done, this one done. But if you say DT, it is 3 line. Okay. So that's how the program is done in a way. What should I do if I get one more DT? So the array works, it add more data and then put it in a longer structure of data, that it is DT. And DE means it is in 2 line. OC is taxonomy, it's 3 line. So I need to collect all the 3 lines together. And then what I do? Then I break it and know these are the taxonomy. Then I put it in catalog. And also I count it, because I need to know how many times each taxonomy is repeated within the total data. That gives an idea. And once the catalog is built. So these are simple things, not very challenging, but we need to know how we do it. Is there anyone who want to start coding this? Any of you have installed this IDE or Python or something? This is actually Anaconda. Anaconda has lot of together. See, Anaconda is like a complete navigator where it gives all the like Spider, or we can have our studio also here. It's all integrated, so we don't need to install each of the software separately. So this gives an easy environment. So now we are using the Spider, which is easier to write and check same time. So anyone want to write, do coding?
Like now, sir, you asking now to code in front.
Yeah.
Sir, this is difficult for us now because we don't know this. Even I faced problem in installing Python. It's
Oh,
problem.
no, that's why I say you install Anaconda, because you better go for integrated platform like installing things rather than go only Python, right? We don't know which one is what are the sub data or things are required. So rather
Sir,
than
how to install that? I don't know, sir.
no, you just go like Anaconda. You just type Anaconda Navigator installation, then you will get for Windows. Because this is like a, because sometime what happens, like whatever software we want to install, there may be some common between like our studio and Spider and things like, they may use some portion as common, but when we install separately, right, there will be some problem, mismatch will happen. So that's why we suggest to have a integrated package like Anaconda that comes in a better way, that it can help faster, even it has lot of way how we can use things, because this is, they are adding more. Orange is also one of the like machine learning prediction and things, data visualization. So earlier it was very few, now they are adding more. Spider is the one which I was showing. So like that they are adding more thing. The good point is the common backbone is same, so that it doesn't add more space to install individual software, and it works very fine, so it is integrated. Because earlier also I tried to install Python, then some libraries doesn't work. It get into a trouble always, because we don't know whether it is able to add or not. But integrated this kind of thing, it automatically does, because periodically we can say upgrade. Then see in the button it say upgrade now. Okay? It means it will upgrade whichever software update is required. Okay. So the, what is the concept for coding? Anyway, don't bother. It is like a big coding class. So what we say, read the input file, right? Name the input file. What is the input file? And then load the file into memory, and then read the file. So here comes the question is, if it is I have to read, then it will become like I'm reading in. So it is like a paragraph. I read the file. So one by one. So for example, if I want to read this file, I will read first this one, right? So I read this one first. I read this line first. So this gives an idea what is there. So first line I read is this one. Second line is this one. As I mentioned in the paragraph. So what it tells, if I read this, if I, the line start with ID, then I do something. See, that's what this line is doing, right? If it is start with ID, then I am adding data into this. Very simple. I stripe all the unwanted character. It's only 2 lines. See? It's only 2 lines. I put extra checking, but it's only 2 lines. So, and then next paragraph is easy. So it grows like this. So you have to practice 2. Only 2 we can practice. Then you know, I can read it. I can write it. So it's not a big challenge. Even if you can do this, then we say, I can write it. 3 lines. 3 lines can grow any number of lines once you have confidence. But do it in a proper way. Put everything in paragraph, like so that you know what you are reading. So I always say I put the example here and then I check, because how to code is not very difficult one. You ask Google, it will tell how to match the beginning of the string. It will give you a line of example. So you can cut and paste. This is how we build. Not necessary that you remember everything. No need to remember coding. Only thing is you need to know how logically you have to build your program. That is more important, because that Google cannot tell you. How I go through this file, how I structure my coding, then what is my loop. See, if I read this, it will end the, which one I will end? Can someone say? So this one set from 13 to 23, one set, how it will complete? Actually, just now I showed you. Anyone? It ends with sequence, right? Very simple. I just now showed you. It starts with ID, the ID in the database, accession number, and finally with sequence. See, you can see it starts with the ID and ends with sequence. So the last thing I need to pick up is the sequence. See, sequence is here. So once I complete sequence, then it tells me here is the break. So once I reach this, I can cut and paste and say, once I reach this, it's a ending. So once I reach, then it will tell me, right? It's the end of one protein. So I can say one protein record, I got it here. So then I can break it, right? Whatever way I want to break it, where I want to put it in array or memory, I can simplify like memory place, I can then later process it. So this is a one set. You can say read the input file and take one set of entry until we get the sequence. This is a terminator they give, 2/. It means one protein is completed. Then I go for next protein. Means I am going for the second record in my database. So this is how we categorize and write up the code. Even if you write these 3, then I'll be very happy. I say, okay, I can read the file. I can load the file and read the input. And I found the ID is the first one. It is telling me that ID is here. See, once it say, it will print here. I got ID. I got the ID. It will say input means it will stop until I say go for the next line. Is it clear? Coding is like building
So can you please show how you input the file?
Yeah, you ask Google. You experiment with Google. Because each part, I told you, right? Logic is your brain. How to write the coding is Google brain. You take copy from Google, cut and paste. We also do. We also sometimes we ask Google and it give a sample. Then we now Chat GPT also there. If you give Chat GPT, I want to read this file, it will give you a code in Python. So not that we need to be a very classic programmer in writing. Only thing you need to apply your brain or idea, logic, how you divide it. Then it's because Google cannot tell the Terminator and then what you want to do. So step by step you get an idea and then build the thing. See, here we read the file. Read the file is very easy. For we read it each line from the memory array and then we go through each line. So that is not a big deal. It can be done in a different way. Everyone has their own style how they read the file and process it. They put it in memory or read it line by line or things like that. They put everything in a big memory and then read it. That will be much faster based on the available memory and the size of the data we are dealing. Because these are all data small, so it can load it entire thing in memory and process faster. But if it is a very big data, then you need to read the data actually 100 lines or something like that. So otherwise the memory can crash anytime. So all this logic you have to understand what big data you are handling or very small data you are handling. So accordingly you have to build up the coding methodology. Is it clear? Yeah. You try, okay, no problem. We can have classes. I think Python classes are going on in parallel, right? Some classes are going on. I can also take some classes when I get time. And we go by example, step by step. So we take these cases, how we can break this big NIPROD data. Or even if you some people are challenging, you can take NCBI data, which is more challenging, but still it is possible. It's not like beyond our logic. I actually done coding for NCBI also, even though it was quite challenging because we don't know where it start, where in, but still as a human how we read. So we have to see as a how the machine will know like a human. Then it is possible for coding. It's not a big challenge then. Yeah. Okay. So this one. So these are the way we break it and then put it into the data, and then data is like a table, and then the table we have a couple of tables for each web server, and then launch it in the web server with front end tools like we use HTML, PHP, and that brings data quickly from server database to the front end. Okay, we take 5 minutes break. Then we go for the Weka. I think some of you are still installing the Weka, right?
Yes, sir.
You can also install this. Yeah,
Sunil,
Anaconda.
if you want to give 15 minutes break, then I can take over, if that is okay with you.
Yeah, that's good also.
Yeah. So is it
Meanwhile,
fine, everyone?
they all can install the softwares.
Yeah.
If anyone want to have a look on Anaconda or anything, yeah.
Yeah. Is
Later
it
they
fine,
can.
everyone? Yeah.
Yeah.
Yeah. So friends, this is what we got. What we needed to do was I needed to run ISAC2. I ran this particular ISAC2 here. Okay. And the data set was here, and we have used that particular paid in dataset collection. We gave 118, and here we searched Homo sapiens, right? IG38. Okay. Yeah, 1 minute. I'm sorry. How about now, Goldie? How about now? Better or?
It is okay, sir.
Okay.
Sir, actually, we feel like you're far away from mic or something like.
Yeah, just give me a minute. I'm sorry. Just give me a minute. I'm using my what do you call? What is this called? a web camera, which has become old, I think, just like me. 1 minute.
So you are not old.
How about now?
Very much clear. Yeah, very much clear.
Yeah, good. So I'm using my this thing. So what I thought was, yeah, I hope my screen is visible. Yeah. So so this is what we have done. We have used in built-in genome NG38, and we ran HiSat2, and we got HiSat2 results somewhere here. Okay, where is HiSat2 results? Yeah, this one, HiSat2 on collection, because we have used our dataset collection. Okay, and this is what we got for individual datasets, which means for both control and treated, HiSat2 was run. And in order to see the HiSat2 results, okay, you can see by clicking the results. Okay, 1 minute. Okay, yeah. So this is the result. It will open up here. This is how you see the HiSat2, and the final summary. How much alignment is done. That is also shown here. Actually, okay. When you click it, usually you get the result. 1 minute. It's not coming up because it's a collection that we have shown. That's probably the reason. It will tell you how much alignment is done. Actually, okay. So this is what you get to see it. Okay. I'll show you. It will come up because it's still loading. It will come up, and then we have this BAM file. You can, if you want to run SAMTools sort, you can run it, or if you want SAMTools mpileup, that's fine. Okay, but earlier, I haven't also used Trimomatic here because Trimomatic is known, and if you want to use that, you could use it. But the very first important step that you could do is cufflinks. Okay, cufflinks. We are using a cufflinks cuffdiff pipeline. So run simply cufflinks. Okay, where for individual control and treated sample, it will measure the transcript abundance. Okay, so since it's still loading, please have your patience. I already ran it for you. Don't worry. Okay, I'm just going to show you how I ran so that we can save some time. Okay, so click cufflinks. Yeah, here you get. And what was the thing? The result was HiSat2 126, isn't it? So here you can give copy link. Here you could use 1 minute. Yeah. Okay. And here you could use 126. Okay, number 126 here. That's not coming because you have to click this single file. Okay, and here you will get 126. Okay, this is a batch mode input, which means separate jobs to be triggered for each dataset collection because it has both control and treated. That is how we have loaded right from sequence to data archive. That is the reason why you do this, and you click run tool. Okay. So when you do this, you will get these so many cufflink files. Okay, you say cufflinks on collection 118, alignment reads, gene expression data for individual. Okay, transcript expression. Okay, assembled transcripts and total this thing, and you will get individually for both the data sets again, for both control and as well as treated. Okay, because it's a data set collection. And now, if at all, if individually, if those data sets were replicates, you would probably use cuff merge. Okay, but here we don't have any replicates, so we are only sufficing with cufflinks. Okay. Now, next one is you go for cuffdiff, the final, differential gene expression analysis. Okay. And here also you click this, and you are using different options here. The very first thing that you use is you use transcripts, the assembled transcripts as an input for one of those particular files. Okay, and then you use the condition name. This is a very, very important aspect here. Okay. So what is the condition? The first one is, okay, you select this. Okay. The first one is you take the condition name to be control or normal. Please have some patience. It takes a lot of memory intensive thing. Okay. This is what you need to take it. Okay. Sorry, where is it? Yeah, cufflinks this. Okay. Yeah, you want to take that assemble transcripts. 1 minute. Assemble transcripts. Okay. And our input data is SAM or BAM, isn't it? And then you type the condition name. Say first you always give control against the treated. Okay. So give control. And then you click this. That control file is this. That is the collection, right? Okay, that is what you give. And the condition 2 is the treated. Okay. And the treated also you give this and again give that. But here, you should be able to select the other file, the other specific SRE ID. Okay. Because you have run it as a collection, because you have used from SRE, both control and treated condition, you have to define it, control and treated. Okay. So here you will be giving the transcript file, and here you will be giving the assembly files in the form of, SAM or BAM. Okay. And then click run to. Okay. It will take some time. If it is not able to split, it will take some time. Okay. It will take some time, and then you will get the results here. Okay. So this you need to give this, and the other conditions, if you want, you can give that. Okay. It will take some time. So let's wait for this until we analyze. Okay. This is what I wanted to tell you. So yeah, I'm sorry, Manav. Now I hope my voice was clear. I've just seen that. Yeah.
Now it's clear.
Yeah. So we'll just wait. It will run in the back end. So we will again see after Sundarda finishes it. In the meantime, let me finish that job. Okay. Yeah. Sundarda, over to you, please.
Yeah, I think now you all downloaded the Veka software, right?
Yes, sir.
So once you have downloaded, you can see a symbol like this. So you get this software. This is a bird. So once you click on from your desktop, you will get to this. Are you able to do this?
Sir, whether this is to be inside the Sanaconda?
Now, after installation, there will be an icon in the desktop which will say Veka GUI. Then you click it, then this will open.
No, ma'am, this is a different software, Vika.
This is a Veka you have to download. Have you all downloaded the Veka? See, this is the, I told no? This
Yes,
is
sir.
Sir, I have installed it actually.
Yeah, once you install, you see this symbol, bird symbol, small icon on your desktop. So if you double click, you will get this window. Everyone got this? Okay, so once you get this, see, you can see it will be like installed in your program. Vika 386 data. If you go to this, you can see this black box, right? Command prompt box. So once you go here, you can see what are the data available. See, we have a breast cancer data, contact lens data. So all these data are clean and tested for modeling, and so we can use it for learning purpose. We are going to load this data, the breast cancer data. Okay, so what we do, we go here and click Explorer. So we open a Explorer where we are loading a input file. So how you build this data in this format, all it will take longer time to understand. So because this data has a different format. You can see when I say the ARFF is a particular type of data format which is usually understood by ARFF, this VECA. But now they can read from Excel also. But this is a original model they give. Like we give all the details, who published it, and then what are the attributes. Attributes is column. So class means it's whether the cancer is there or not, and what are the age group. So this gives a more detail. All these other things like tumor size or what are the things where the cancer was found, and whether it various parameters defined by medical term doctors who are dealing with the what do you call cancer treatment or that domain. They bring out all this information to conclude whether it is going to be a cancer or not. So all these parameters, these are all validated several times. And then finally what we say is class distribution is how the data is taken up. So we have 201 data which say there is no recurrence, means the cancer is not there. But is the recurrence is 85. So this is a cancer and non-cancer data. So total we have 286 data, and attributes we have 10, and these are the data. These are the data. Each one is how it is column is driven with whether character or number or things like that, and then they build like this the relation. So this is a structure how the data is drawn up in this particular format. See, so it gives the data like this attribute. So each attribute, it gives what are the options available, if it is a yes or no or left, all this. So this gives an idea what are the data likely to come here in the below. And then this gives the actual data. So this goes the actual data for each patient, which is how many number? 286 patient, right? Out of 281, we know some are like recurrence and non-recurrence, means it's a cancer or not a cancer. So we get all the data in a classified in tabulated form, and this we can put it here directly. So I say open file. Where I open the file, this one is under the program file, Vika, then I go data, and then I take breast cancer, okay, and then load it. So it loads the data because this is now ready form. And how to build this data, it takes a longer time. You have to do a properly having curate the data and assemble it in a proper way, and then you can have the text file like these files which are going to be with these files are this one is for us is very easy. It is there already, so we can just use this data, and then we can test for our learning. See class, it will tell how many are there. 201 belongs to this class 1, no recurrence. It's a good sample. And this one 85 is the recurrence event, mean the cancer is there. So that's why it is red and blue. So this class, and each one we can know how the data is divided between two classes. class 1 is cancer, yes or no. So see, all this we can see how data is parsed. So this advantage of this software, it gives pictorially everything clear. We don't need to check one by one. So by just clicking, we know how the data is divided for each of the and sizes, squat, irident, everything. All these are medical thing which we don't bother. The idea is how we build the model to know whether the cancer is there or not, right? So then how we go? We go classification. Means we want to classify this data between two segments. One is yes or no cancer, no cancer. So we use an algorithm. So we take any algorithm. There are a lot of, we go naive based, which is a faster algorithm, and then cross validation is whether you want to divide the data in 10 and then rerun them. Like take one as training. Training means I know answer already. I don't know the answer. Like that, we divide the data and then run it. So, and then we run and click. So, see, the 205 and 81 is divided. So, the 71% the data is accurately predicted, and 28% it cannot predict properly. So, the table gives the below how many samples, like no recurrence, 168 is predicted correctly, but 33 predicted wrongly. And recurrence, 48 predicted correctly and 37 wrongly predicted. So this gives an idea with the data how many number of samples are divided, accurately predicted or not predicted, and gives the overall percentage 71%. Okay, this is one particular algorithm. So advantage of this software, we can choose another one. Okay, I want to go function. This is what neural network, multiple layer perceptron, which gives a more layer based thing. And then I go start. This takes a while because it take a model and built the model one by one. So it gives all data how the data is run for all the division. See, it gives various data and what is the level, the prediction. So finally, after all these analytics, it finally gave the. It takes a longer time because so it 180, 150 is accurately predicted by this neural network multi-layer perceptron, and 50 is so this one 168 predicted 148. This one is 150. So 200 is predicted correctly between cancer and non-cancer. So that's why the percentage changes, right? Between this one is 71 and this one is 64. So like that we can run other algorithms also. We can have some support vector machine and then we can run it. So see, it gives 69% accuracy. And how many? 171 correctly and 57 correctly predicted here. Then we can go for other rules, which is a decision table. We can run through it. So this gives 73%. Each algorithm has its own way of dividing the plane. The idea is if we give a big data, how to divide it between cancer and non-cancer. So this algorithm able to divide 73% and 60, but SMO 69 and 30. So each one has its own merit and demerit. But for biological all kind of data, we prefer neural network because which does a better way of handling data and then gives a more accurate level of prediction. But usually we try with various algorithms. J48 also another, this gives 75%. So each algorithm has its own merit because based on the data, if it is only two data, like only two yes and no, then the support vector machine may be a good idea. And if the data is very less, we use multi-layer perceptron because that can build even with small data. Naive Bayesian is a general thing. If you have very huge data, then naive Bayes give a quick idea how the data is parsed, whether it is data is good without bias or things like that. Okay, anyone is started doing this?
Sir, no, sir.
Doing?
No, sir.
Yeah, you are supposed to do this, right?
Sir, not clear.
Parallelly, you have to load the data, right? You have to see pre-process, open the file. We can use another. Okay, I can use class. So how the type of class? There are so many types of class is there, and then how it is all the data divided. Because these data are clean and tested and published so many times. So we can easily learn from this data. Even you can work out all the data, whichever data we have, you can try with all this data. We have all the data, contact lens, diabetic, all the iris. Some of them soybean. Weather, weather is also a very good one to understand whether it's going to rain or not. I don't know whether it will work good for Singapore because Singapore rains every day. So we have to see whether it works or not. So there are two set of data. One is with the nominal. Nominal means we have a different set of names and things like that. That's nominal. But numeric is number based. So they have two set of data. Why don't you load something and see whether it is working? Because just now we see the cancer, right? Breast cancer. Some of you load it? So open file. How you got this? First, initially we come from this, right? This is the first small index. Then you explorer. There are a lot of other things out there. Knowledge flow, experimenter. These are all step by step we have to learn. This one is the first to learn. So are we all in the same page? Again, I open this. Then again, I go class. See all these. I I choose from here Bayesian. I was choosing. Okay, I choose another Bayesian. And then start. Just click. See, it one is 70%. But these are all maybe not accurate because these are all works maximum.
So what's the file format? Index means input file format.
In fact, I showed you, right? This one is a specific format. It works like this. This one is like a relation. Structure is like this. Relation. Every parameter attribute age is grouped like this. So you can see like
Can we load Excel file or any text file format?
Yeah, we can load also. Now, as you are practicing from this, you are going to use the known things, right? That's why for learning purpose. Later, you can load from Excel. I think it gives all, see, it gives all format. You can CSV file also, you can load, data file you can load. See, all the formats are there. From Excel, you can create a CSV file, right? Save as ms.csv and then load it here. There are various formats available here. JSON. This is the one usually the author created. This one, but we can use CSV also here. From Excel, you convert it to a CSV, then you put it as file here. Various format can be used. But for testing practice purpose, use the ARFF which is already there, right? Instead of now trying with, you can use any data you want to use.
I tried with one TSV file, but that is not taking.
No, you have not given header properly, you know. The
Okay,
first
sir.
line should be a header.
Okay, sir.
Because first line should be a header, right? Always a header in the first line and the
Okay, sir.
data in the. That's why it is missing. Yeah, you should know what is the header. See, like all this header, it requires to know. And then comes class. Class is how the data is divided between this is cancer and non-cancer. We have a total of 286 data here. Can any of you try? If you are in group also, you can try together. Okay, I give 5 minutes more, then you check and then tell me. Once you are clear on this, then we can go for a.
Sir, if the file is loaded successfully, whether it will show that file is loaded or something like that?
Yeah, it will show like in this page you will see any file you tell me. See, I'm loading diabetes. See, it loaded without any error. If there's some problem, like when you.
I'm not getting, sir.
Why don't you try with this data available here? Because Vika gives all this data, right? It's easy to, for learning purpose, it's easy to use this data rather than later you can create your own data.
Sir, whether this data is shared already?
No, when you install this Vika, it comes with that. Today only
No,
I installed this.
sir, I have installed it, but I'm not seeing that.
No, you will see this. See, when you install this, there is a directory called data.
Ma'am, it is a preloaded data. You can find it in the installation folder of Vika.
Yeah, where you install. Usually, automatically it installs in program file. But
Yes,
if
sir.
you
Yes, sir.
change the directory, then you check where you have installed backup.
Yes, sir. It is in program file.
Yeah, like same thing, right? Then you go to data. Even you have a lot of document also, how things are working. Everything is given by them because it's a free one. So even you can see what are a lot of documents also. It gives a lot of document, how to read things and then data.
Yes, sir. I'm seeing the files.
Yeah. So you can load any data you want.
In this, for good model, how much that value should be? Hello.
Can go airline also if you want to load airline. Airline is only two passenger numbers and date. Only two. No. I think this what they want to classify date.
that kindly repeat the steps after loading the data.
Yeah, this is a pre-process. I've loaded it. Okay, I go again, load the breast cancer. So, see, it will tell what is the relation, and
Yes,
what
sir.
are.
Same page I am here.
Yeah, you then go to classify.
Okay, sir. And here I have to test option with whether I have to use training set or
Okay, say I removed all the thing done. Okay, I go to here, I go to base, choose the classifier base section and then naive base. Okay, without changing any of this. Later you will see what all these parameter. So cross validation 10 holds. Then I say start. Say it finished this. It loaded this.
it is selecting only 0 or other than that nothing else I could select.
Which one?
In the classifier choose.
No, you go choose. Then see it is like sub selection. You click here the greater arrow. Then you go to the naive base. Select it. Click it. Then you go for say if you click here, it will tell what is these options available here. So when you click here, for naive base, it will tell what are the options available, batch size, various parameters. But now don't bother about all this because this requires a lot of understanding theoretically. That requires a couple of classes to understand what all these parameter is. So that we are not going there. We just use it as a package. Then it's easier for us to move on. So then I say start. So it clicks and run. The algorithm runs with the data 286 and then it finally gave a classification. I mean, how much is classified? Out of this 165, 168 is correctly classified as non-cancer and 48 correctly classified as cancer. And this 33 could not be classified and 37 could not be classified. Because how do we know? We know from the sample, right? See, these are the data. So the class label tells this and this. But some of them could not be classified because the data may be having some problem or not accurate or complete. Always I told, right, we cannot expect to get 100% because the data may not be so clear and accurate. There may be some problem while data collection or cleaning. Some data may go missing also. Many times we go missing data. Then we take average and then sort things. But it may not be accurate.
Sir, I got the same result.
Yeah, you will get same result, right? Because same package, same input. Yeah. So then you go to next one. You can go to function and say multi-layer perceptron and then say start. This will take a while. Usually I go for naive base, multi-layer perceptron, support vector machine, which is SMView, and decision table, which is J48 or these 4-5 are standard we use. See, this one you can get same result. So it gives 64% accurate. And naive base what 71% accurate. It can correctly classify up to this only. This one wrongly classified. Mean the data it cannot classify correctly. Are you getting this?
What we are interpreting from the results? What actually we are doing with this?
No, we are seeing by this algorithm how many of these 286 samples are accurately modeled. I mean the model is built and how many are correctly classified. See from non-cancerous 150 accurately classified, 51 records could not be classified. But still we can go into a one record level to say which are the records 150 are accurately classified. That is possible. But that one in Vika is difficult to do. We have to write coding. We use the Java backend. We can call the Java in the backend and then we can run through sample wise. Then we know which are the samples accurately predicted. That gives an idea. when we build model, which are the 51 wrongly predicted, and what are the problem, whether we can improve the data or clean it up or things like that. So that can improve further.
So whether we
This,
can use this for any genomic data to have this training set and then test set.
yeah, any data you can give. I told, right?
Okay, okay, okay.
Building data is your challenge. What you want to build, the column wise, and finally we can. These are the sample. This is a classic sample used for a longer years to teach how we can use this algorithm and divide the thing, and then understand how it is working. That is the main thing. Then you go for another one. You can say rule based decision table, and then you can trace J48. These are the usual standard. We can use other things also. There are lazy functions classifier.
Sir, I'm trying random forest with this.
Yeah, this is support vector machine because these are the standard thing we use: decision table, multi-layer, it's a neural network, naive base. J48 is also one of the decision tree, and SMO is the support vector machine. So these are the 5 standard algorithm we use to understand the data, how it is parsed, whether the data is clean or we are able to build anything with this data. So all this we can have an idea.
Sir,
And also you can play with, see, you can play with this. I want to put cross validation tree, then what happens?
Sir, we have given cross validation of this one. That means already model is developed for this.
See, you can see when it was 10, now I changed to 3. See, there are a lot of changes here. From 171.57, it is changed to 158.54. So it changes because the number, when I divide it into 3 portion, it is not that accurate. Means some portion are changed into the other side. So it's wrongly predicted. Here it was 171 because I divided in 10. Now I divide it in 3. The entire data cross validation divide 3. 3 set rather than 10 set. So these are the parameters you can play around, but some changes will be there. Then we can see which portion of data is not accurate. So that helps in, but we are not going analyzing all the data portion, right? So we want to understand how much we can build that level of accuracy. That's the thing, and whether we can improve it, and then we go for prediction. So far, we are not doing prediction. This is building a model. So can we do a prediction? Means you build a model and then we can predict it. So give some portion as unknown. You don't give the name because now I give it right, the label. Yes or no, cancer or no cancer. I suppress the label and then run it. It's possible to run and get what is the answer. So that will give, you should give exactly what is the answer. That is a larger step to get the answer. That will tell whether you give a new record. It will tell whether it is cancer or not cancer. Right? That's the ultimate idea. Before that, we see whether the model is accurate with the data available. Okay,
Sir,
any detail?
sir,
Yeah.
yeah, you told about neural network, which is fit for biological data.
Yeah.
So whether in this option neural network based is anyone?
Yeah, this is the neural network, multi-layer perceptron.
Multi-level. Oh, thank you, sir.
Because this software, people, they named it as multi-layer perceptron. I mean, neural network is a multi-layer actually. It has more layers in between, from the input layer to the output layer. So they called this algorithm name as multi-layer. SMU also support vector mission, but they put it as SMO. It's that style of putting, but they in the detail we know it is same as support vector mission. So the classification is clear. You can do practice. Later you can come back. Maybe
up to this we are clear. Means we have done cross validation. That part is we are clear.
Okay, then we go cluster. What is cluster? Means if I don't have a label name, how to divide the data? So I want to divide the data. See, the clustered data is we have it divides in three parameter. So we had two, right? We had two. One is cancer and non-cancer, but it says there are three cluster. Means one is yes, one is no. There is another third one which we don't know what it is. Means which are not connected with the other data properly. So cluster gives an idea how the data is divided. Means if it is two, it should have divided two. Now we can see that 65 is divided in a separate. That is the reason we can see here also we get some noise. See, we get this noise because this one is not connected. The data is not clearly connected with the other data for each of the attribute. That's why we get a third cluster, which we are not supposed to get. Right? These two data represent in the classification also same. But this one comes here as a cluster. So cluster gives an overall idea how the data is divided.
Whether this third group or third cluster explains that there is no correlation.
Yeah, the third cluster is the same which they are unable to allot the yes or no.
Sir, this third cluster means now it is showing that data is from training set, use training set data. Where it is defined as the data is a training set or test set data. How we are defining. If we are using our own data, where we have to define that this is.
No, we can. I told right here also we can use training and supply test data. This is where I want to test actually. I told right we can give some portion for testing without label. So I can divide the file into two, and then I can use training the whole thing, and then this one I have to give a test portion without label, and then check whether it is getting all the same data.
Means test set is without the header.
No, header should be there. Only thing, it will not have this one, this class.
Okay.
Whether it is cancer or non-cancer. So
take
some portion of these two, remove the column, 10th column, and then give it as a test file. So you can supply a test file here. If you click it, will ask where is the test file. You can add it. But this one you have to build on your own. Maybe you can use Excel, load it, and then take some portion. But one important thing is don't put in both training and test. Right? Then what will happen?
Sir, if I'm deleting any column of the training set,
not column, row.
Okay. So
all columns should be intact. Don't remove any column because column should be all same, all 10 column. Only thing this for test file, this 10 is not there. Simple.
Sir, whether it can be anything or only the class.
All columns should be same. Only in test file, this column 10 is not there. Because that is the answer we are expecting.
Sir, I couldn't I couldn't convey my question clearly.
Yeah.
My question is always whether it should be the 10th column or we are defining the 10th column as the classifier here.
No, the thing is, idea is anything named as CLASS attribute, then the software understand that is the answer.
Okay, sir.
But usually we put it in the end, so that we know the flow, right?
And there is no need to have always the 10 kind of thing. Means we can restrict that with 9 or 8. whatever.
Yeah, we can rebuild also. See, we can rebuild this like this. Okay, then
Okay.
we can download. See, we can remove. I can
Okay.
say
Okay.
some column I don't want to add. Then I say maybe this one wrong, this one wrong. So I just remove it. We
Okay.
can just remove it. It is not removing from the file. And then we can still build with this and see whether anything is improved. Say same SMO I want to run. Let it be a cross validation. And then start. See, it has increased. See, it has increased. That means those two column may be not accurately giving data. See, it has increased to further 175 rather than 158 here. See, both these are increased further. So that is how we play around. We see whether some of the column or may not be contributing better to the model or which is giving a noise. Sometimes it gives a noise and it degrade the percentage or accuracy. The software advantage is you can rebuild, you can save it also. You can just save this small file as a new data set and then you can process it.
Clear, sir.
Yeah,
Understood.
yeah. Cluster is we can still run and see whether what happens to the cluster. It is changed.
Sir, I just wanted to see one model development.
Yeah, the model only we developed here, no? All this model.
Okay, this is called model. I
Yeah,
thought
this is the
model
model.
means we will have one linear algebra formula kind of thing, something like that.
Algebra.
No, sir. Model means like regression equation. We will develop something. I thought like that.
No, 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 no. This model is a classification model, prediction model. Means we build and see whether a new sample is cancer or not cancer. This is a prediction model, not a mathematical model. It's based on
Okay.
mathematics, but not on mathematical algebra model or something.
Okay, sir. Okay, sir.
This is a prediction model. Okay, then cluster. Then what is associate? Associate means we can see how it is associated. See, the variables, how they are associated. It gives an idea. See, age 50 to 90, and then it is like a mathematical way of correlation, like how things are attributed or defined, or it gives an idea how, if they, we say, no, right, 50 to, if the age is 50 to 59, if the node is 0 to 2, class is no occurrence, then it mostly the answer comes like this. So this given association of among the variable and the value, how it is associated. It gives an idea what are the important variable, how it is interconnected. So the associate rule gives an idea to know how things are happening. Okay, we go here and load the total file because we removed some portion. So we reload. Okay, what is the next thing is select attributes, because here we have so many attributes, right? In this another sample diabetes. So select attributes can check which are the attributes which are good. See, now it says 2, 6, 7, 8 are good. Means you use only these sub from the mean. See, instead of using everything, you use only this 2, 2, 4, 2, 6, 7, 8. Right? 2, 6, 7, 8 and class. That is enough. So other things we can delete also. That's what this select attribute suggests, that these are all the contributing attributes or column for this model. So this gives an idea in understanding which are the attributes good for the model. These are a lot of other things which we are not going in detail. There are a lot of there are algorithms in this. We can use so this gives everything. All are good. So it depending on various info gain or classifier. Correlation also is good. But it is also almost, see, this gives an order. You can see how the order means 2, then 6, 8, the weightage. The weightage is ranked like this. So based on the ranking, it gives, you use first 2, attribute 2, then 6, 8, then 1, 7, 5, 4, 3 in this order, because this is the value for each of the attribute. Rank means it brings the rank how to use. If you use only this and you can do a good modeling, then you can say. But sometimes experts say this is an important variable, attribute you must use. That you have to consider and get a cleaner data and use it. So always don't go for the prediction model. We also take expert advice in knowing which are the important parameter attributes medically, so that we should try to add them. If you avoid them and then say our model is good, then they say no, the important thing you missed, right? So that's the thing. And visualize is usually how the data is visualized. We can visualize between, say, various attribute and we can play around select attributes or which are the thing, how to represent. We can have a vision how it is represented by class.
Can we download this image, this plot?
Yeah, you can take a picture, I think, and download. I think you can download. We have to see how. Or easy thing, you have to take a picture. You can take a screenshot and
crop it.
Yeah, crop it and cut and paste.
Okay, I'll try.
So any question on this select attribute association?
So this select attributes actually, like, we couldn't get what we are exactly doing here.
No, see, we started with all these parameter, right? Attribute
Yes.
1 to 9. So why the model is not getting value properly? Why there are some lagging, right? Because some attribute may be not contributing to the model. So this select attribute will try to run and see whether to build a model only this 4 is enough 4 attribute is enough. 2, 6, 7, 8. So if I go back, this 2, 6, 7, 8. These are enough. The remaining thing class is required. The remaining is no need actually. According to this algorithm. I told right, but we have to check the expert opinion whether anything else we are missing. Right?
Sir, how you have identified these attributes are needed, others are not contributing?
No, this running this algorithm based first CFS subset. So when we run this on this data set, it will decide for the column. These are mathematical calculation. It finds only these 4 is enough to get the value of the model.
Okay, here with the selected attributes. Okay, then again with the selected attributes, we have to run the first part.
Yeah. No, you go to the, see, 2, 6, 7, 8. So, 2, 6, 7, 8, right? So, what I can do here. So, I remove other things. So, I keep 2, 6, 7, 8, 9. So, I just remove this. So, this is parallel to what this gives. Place, mass, PD, age. See, these 4. And then I try to run the same thing and see whether I get a better. See, it gives a very big difference, right?
Yes, sir.
So that's the thing, because select attribute can tell by statistical or correlation thing whether it is, which one is enough to get the column attribute and build the model is enough. But again, I'm repeat, if anything we miss important to the medically some of the parameter that we need to check whether the data quality is good or bad, that we have to re-include, because we cannot skip what expert says. Like sometimes they say clinical assays or some X-ray or for the cancer they might have having a image analysis. That is more important. That we cannot say that data is not clean, so we can just remove it. We cannot remove it, right? That's an important thing we should remember. But as a software level, it gives an idea what are the things doing good. Then we have to analyze it, whether these attributes are good enough or not.
So,
Okay. Yeah.
So at the end of this task means if I have found some say model which are perfectly diagnosing the things, no. So means how could I use that particular model by hitting the query, single query to this software?
Yeah,
only?
that's what I told right here. See, this one is whatever data I have. Okay, we go to the earlier one. So out of this, I take one or some data and put it as test set and remove them from the training set, and then use the training set and supply this test set. That 3 will come as answer. What are the answer is going to come.
Okay, sir, got it.
So that's how we build up and escalate the model. Not only that we build, the next step is to predict, right? So you take some portion, but caution, don't keep in both training and because if you give question paper with answer paper, then everyone will get 100%, right? Same way, and if you give training set, same sample you use for test, then it will become 100%. So be cautious, remove from the training set those data and put them as a new test data and then run it. So each line it will give what is the answer. You can check whether it is correctly predicting or not. Same if the sample belong to this portion, it will be wrongly predicted because the data is not clean. If it is here, it will predict correctly. Okay. Any question?
Sir,
I think.
Sir, it is very clear. We don't have much questions now because we will try this, and if we are having questions, we will get back to you. But we have one request. Yesterday we had one lecture, and there feature extraction was told. So, if we can get some hands-on experience in feature extraction. it will be for us.
For yesterday lecture, I have no idea. We have to ask the lecturer or Prashant.
Sundarda, it was Jeraman sir's, simplest libsvm actually.
Okay.
So maybe you could have probably you could ask them to use Vika and run SVM. SMO model you already demonstrated, so that can
Yeah.
be given as an exercise. Yeah.
Yeah, you can use it. I told right, as long as you have the label or unlabel. If you, because label means it's supervised learning. Means I know the label. Unsupervised means I don't know what is the answer. Then we can use cluster, which gives an idea whether the data is can be divided into two pieces, good and bad, whatever it is. So that will give an idea, overview how data is good enough to move on for modeling. You can use it, yeah, the yesterday sample.
Okay, sir.
Okay, then if not anything, I think we have come to the end of the session. Anything you want to add, Prashant?
No, sir. No, da. I mean, accepting that I've added a kind of a exercise for them in the online